Hello, and welcome to the first of our two planned Spectronaut user seminars at this year's ASMS. My name is Tejas Gandhi, your host today, and just a little bit about myself. I did my PhD at the University of Groningen in Netherlands, and I've been part of the R&D bioinformatics team here at Biognosys for the past several years. I'm very excited about our lineup today. While it was not exactly planned to be like this, but both of our presenters will touch on the topic of doing proteomics with the latest yet different ion mobility-based technologies. Certainly one of the hot topics at this year's ASMS. Also, it is very interesting for me, as our team has invested a lot of effort in supporting ion mobility data and the latest version of Spectronaut. So first up, we have Jesper Olsen, who will be talking about FAIMS, doing DIA with FAIMS. Jesper is a full professor at the University of Copenhagen and vice director at Novo Nordisk Foundation Center for Protein Research. He has made several important contributions in the field of proteomics and high-resolution mass spectrometry, especially for quantitative phosphoproteomics and its biological applications. One of my first interactions with Jesper and his group was at ASMS 2018, so around 2 years ago. It was at the end of the day in a rapidly emptying poster hall at one of these huge ASMS poster halls. Both of our teams had gathered at a makeshift table between one of these many poster rows to discuss the problem of PTM localization in DIA. This led to a very fruitful collaboration on our side, led by Lucas Reiter and Oliver Bernhardt, which ended up producing an impressive PTM localization workflow in Spectronaut and a Nature Communications publication. Before I hand the floor over to Jesper, I have a request for you in the audience. You can and are in fact encouraged to ask questions. Little snafu. You're encouraged to ask questions throughout the talk, so we'll make it logistically easier instead of having all the questions come in at the end. So I would highly recommend doing that. So with that, I would hand over the microphone to Jesper.
Yeah, thank you, Tejas, for this kind introduction. And let me see if I can share my presentation here. Yeah, first of all, thanks for the introduction and thanks for giving me the opportunity to show you some of our recent data that we have collected on the new Orbitrap Explorers 480 system, where we were fortunate enough to be one of the first labs to beta test this new instrument. So all the data that I'll present today has been collected by Dorte Becker Jensen and Anna Martinez del Val from the lab, and as part of this collaboration with Thermo Fisher. The Orbitrap Explorers 480 is this small benchmark instrument that has the same scanning speeds and resolutions available as on the Q-Exactive HFX platform, but much higher robustness. And one of the advantages of the Explorer system is that it also interfaces well with the FAIMS Pro source that basically allow you to do differential ion mobility between a high field and a low field. The advantage of this is that using different compensation voltages, one can then basically do kind of a gas phase fractionation of the analytes that are eluting off the columns. And as you may be able to appreciate, depending on the compensation voltage, you have a quite significant change in the charge state distribution of the analyte, so you can very nicely discriminate, for example, singly charged background ions from multiply charged peptide ions using these different types of compensation voltages. So For all the data that I'm going to show you, we've used this sample preparation method called protein aggregation capture, where we use either organic solvent, high temperature, or salt to basically precipitate proteins out of solution directly on microparticles. And when we use magnetic particles, then we can very effectively concentrate them on the beads and do very efficient digestion and basically buffer exchange. And we've been able to demonstrate that we can do this completely independent on the surface chemistry of the beads, and this allows us to do really multi-purpose proteomics workflows. One of the advantages of using this type of pack digestion is that we can significantly reduce the number of miscleavages otherwise observed typically in our in-solution digestion, and thereby also reduce the amount of trypsin and lysine needed. And more importantly, we can basically automate this whole digestion procedure in the Kingfisher robot using the magnetic beads from Resin Biosciences. So when we got the new instrument to test, we immediately set out to see what benefits FAIMS could have for our DIA workflows that we've been very excited about over the last couple of years, and where we typically would collect a very deep spectral library using offline high pH reverse phase fractionation and max quant analysis in order to generate a high quality library, and then do the single shot DIA on the Orbitrap systems. And we could see that when we combined the single compensation voltage FAIMS with DIA, that around an optimum of minus 45 volts on the compensation voltage, we were able to identify more proteins and more peptides than without FAIMS on this new system. And most importantly is, of course, the fact that we see that we also get better quantification between replica experiments. So with these new settings, we can basically, in just 21 minutes of analysis on the EvoSep1 system, using a simple single compensation voltage, we can identify more than 5,000 proteins and also have really high reproducibility on the quantification. So all of these analyses were done with the Spectronaut version 13, and now with the new version 14, we were fortunate enough to get early access to test this version, and we were very surprised to see how much we're actually able to increase the coverage of peptides and proteins. So this is, for example, and some of the short 5-minute gradient analysis that we've done on HeLa lysates, also published in the Explorers paper in MCP, and we can see now that if we use the new Spectronaut 14 version, that we can basically increase the number of unique peptides by 45%, going from 8,500 up to more than 12,000 unique peptides, and also increase the number of protein groups from just below 1,700 to more than 2,200, simply just by using the new version of Spectronaut. So we're obviously very excited about this. One of the things that we did in the publication about the Explorers instrument was to also directly benchmark the TMT workflows with the DIA workflows, and we were able to demonstrate that we can basically get the same coverage and quality on the quantification using DIA as one can do with a TMT-based workflow, but significantly reduce the time needed for the analysis. The only caveat here is that one would have to collect a really high quality spectral library in order to get the DIA to work really well. So what we basically did was to go through 12 different tissues from 3 rats in triplicates, and then collect 46 offline fractions. And for each of these tissue proteomes, we in the publication searched the data with max quant, but afterwards we also decided to test the similar performance in SpectroMine. And now with the new version 14, we can basically see that we're able to significantly increase the number of modified peptides identified, the number of proteotypic peptides identified. And most importantly, we can see that the analysis time required for analyzing a proteome, actually searching here with many variable modifications, is significantly faster with the new versions of Spectronaut. So this, of course, gives us a lot of time to do many more searches at the same time. So the most of the data that we have collected so far has been on this 21-minute gradient on the EvoSep system, but we've recently also been able to optimize the 44-minute gradient such that you can generate 30 samples per day. And with that, we decided to also try to switch between different CVs on the fly, or do so-called internal CV looping of the during the runs. And of course, for that, we have to do a number of compromises, that when we switch between 3 different CVs on the fly, we would have to cut down on the number of windows and the window widths for the DIA scan cycle time. So where we would normally have roughly 15 Dalton windows with small overlaps with single CV analysis, we can see that when we go to 3 CVs, then we have to broaden the windows up to 43 Dalton and only go through 16 windows for each of the 3 CVs that we are switching between. But surprisingly, it actually seems to work really well now with the new Spectronaut version that is able to handle these multiple CVs in the same file to an extent where we're basically able to increase both the number of proteins, precursors, and peptides using a combination of 3 different CVs. So here we use CV 40, 55, and 70, and we can see that we can identify in the order of 40,000 unique peptide sequences, more than 60,000 precursors, and 5,500 protein coding genes. And more importantly, when we look at the data from the 44-minute gradient, we can see that we are now up at close to 7,000 proteins coding genes that we can cover in a single experiment using these 3 different CV values on the fly, and close to 70,000 unique peptide sequences and 90,000 precursors that we can cover in this short period of time. And again, we can see that the quality of the data is really high, so the coefficient of variation between replica analysis is actually maintained also for the 44-minute gradients, so to an extent where more than 5,000 proteins can be quantified with a CV of less than 20%. And as Tejas already alluded to, we are of course doing a lot of phosphoproteomics in the lab, and we have recently also optimized our workflow in a way that we can combine the packed digestion with the phosphopeptide enrichment directly in the Kingfisher robot, and do single shot analysis with the EvoSep system that allow us to get really high throughput. And compared to the previous generation of workflow, where we used in solution digestion, titanium dioxide, and the EC1200 system, where we will only be able to analyze 45 samples per day, we can see that now with a throughput of 60 samples per day with this new workflow, we can actually significantly increase the number of specific phosphopeptides and localized phosphorylation sites from these, in this case here, DDA analysis. But more importantly, we can actually transfer this directly to a DIA workflow as well, where we recently, together with Spectronaut, published this manuscript on how to do rapid and site-specific phosphorylation profiling using data-independent acquisition, both with and without spectral libraries, which I will also get back to. And we can see that we can maintain the quantitative accuracy also in DIA, but in DIA we're actually able to cover many more phosphopeptides between replica runs. whoops. And the main reason why we think we can cover many more peptides in this relatively short period of time is because we are sampling many more ions in the MS-MS spectra compared to when we do the normal DDA type of analysis, where we kind of limited in the number of precursors that we can isolate and fragment in a standard run. And an important thing is, of course, that we're also now able to confidently localize the phosphorylation sites in the DIA workflows using this very nice algorithm that Oliver Bernhard from Biognosys was spearheading the work on. And we were able to benchmark using a synthetic phospholipid library and demonstrate basically that we're able to do the localization as accurately as one can do in a DDA analysis using max quant, and actually at very low level phosphopeptides, we actually seem to be even more sensitive and can identify and accurately localize more sites than what is possible from a DDA analysis. And what is now really exciting is the direct DIA that we can use actually without having to need a spectral library. And we can see that there's actually been quite dramatic improvements in the new version 14 of Spectronaut compared to version 13. So we go from something like 15,000 localized phosphosites in a single analysis of 21 minutes to close to 20,000 localized sites. And this is actually even more than what we typically get using a project-specific library. So it seems like direct DIA is working even better than spectral libraries these days. And basically to test and see if all these additional identifications are actually trustworthy, we overlapped the data sets from the direct DIA analysis in version 14, and were all the sites that we could identify in at least 2 out of 3 replicates, so more than 16,000 of them. And we could see that it was only 900 of them that actually were not detected either in our spectral library or in the Phosphosite Plus database, or in this recent large-scale PRIDE database dataset from the Ochoa lab. So we could basically see that 95% of the sites that we identify using a direct DIA has already been described in curated databases or have high-quality data-dependent acquisition-based evidence. One of the things that we've used the DIA workflows for is really to do large-scale interaction studies to study the EGF signaling pathway. And we have recently extended this to also study not only the interactions in the EGF signaling pathways, but basically the signaling in time and space. So to record the spatial-temporal signaling dynamics. And the way that we do that is basically to follow a subcellular fractionation workflow where we, using different detergents, can enrich for different subcellular fractions. And starting from a single P15 dish, we can then use 5% of the extracted proteins for total proteome analysis and 95% for phosphoproteome analysis after titanium IMAC enrichment. And all of these data are then analyzed with the 60 samples per day method on the EvoSep system, and using either library-based approach for proteome or direct DIA for the phosphoproteome analysis. So basically within 4 hours of analysis, we can then per time point capture both the changes in the proteome and in the phosphoproteome. And just to validate the experimental details, we can see that doing, in this case here, 5 replicates for the sample preparation, that we have very high reproducibility for in the different fractions, and overlaying known subcellular markers, we can see that fraction 1 is highly enriched in cytoskeletal and cytoplasmic proteins, and whereas fraction 4, for example, is enriched in plasma membrane proteins, and fraction 5 in chromatin-containing proteins, and fraction 6 in nucleolar proteins. And we can use this then if we stimulate cells, for example, with, in this case here, EGF, where we used 5 different time points, collected 4 different biological replicates, analyzed these 6 subcellular fractions, and analyzed both the phosphoproteome and the proteome, so altogether 240 samples, which basically only took us 4 days of mass spec analysis time altogether. And this basically allows us to look at how the EGF signaling is activated at the plasma membrane, and how that then starts a signaling process that leads to recruitment of adapter proteins from the cytoplasm and starts to signal downstream, and also internalization of the receptor and degradation eventually. And we can basically follow these recruitments to the receptor by looking at the changes in the subcellular localization of proteins in our data set. And for example, we can see that, for example, focusing here on the 3 of the most canonical adapter proteins, that the majority fraction of these, for example, in this case here for Sybil, is before stimulation is mainly in the cytosol, but already after 2 minutes of EGF stimulation, we can see that it changes from the cytosol to the plasma membrane fraction, where it's being recruited by the EGF receptor. And then it basically, over time, changes again from the cytoplasm and back to the, or from the plasma membrane fraction and back to the cytoplasm. Because we also recorded the phosphoproteome, we can look at all the individual phosphorylation sites on the EGF receptor and see that the tyrosine sites, for example, are mainly phosphorylated at the plasma membrane, and they have very rapid kinetics, whereas, for example, serine phosphorylation sites are either non-changed in the first couple of minutes, or they have more delayed kinetic profiles. And most importantly is that we can actually look at these adapter proteins that are being recruited to the receptor and see that for example, here in the case of SHIC1, that it's only the membrane fraction of SHIC1, the one that's actually being recruited to the plasma membrane, that becomes tyrosine phosphorylated, in this case here on tyrosine 427, that is known to link directly to GRAB2 and downstream MAP kinase signaling, whereas the fraction in the of SHIC in the cytosol is largely unphosphorylated. So this is a very nice way to directly look at the spatial-temporal dynamics of EGF signaling. And with that, I'll just end up with some of our ongoing work and future perspectives. So we are now also looking at different complementary compensation voltages, FAIMS for fractionate phosphoproteomes on the fly. And obviously, we're very excited about the spatial-temporal profiling of the proteome, where we can do high throughput workflows now to really comprehensively look at the subcellular localization and movement of proteins. And we're analyzing multiple experimental conditions now in order to describe protein relocation in a dependent manner. And with that, I would just like to acknowledge the people that did the work. So this is mainly work done by Anna Martinez Del Val and Dorte Becker Jensen with help from a few other people in the lab. And then we also like to thank our collaborators at Thermo Fisher, Evocep, Resin Biosciences, and Biognosys. And finally, I would like to thank you for listening in today, and I'll be happy to take questions. Thank you.
Hey Jesper, thanks for the great talk. It's always really exciting when external people find the same results as what we hope people will get. So that's really good to hear. Yeah, so I actually had a question before maybe I announce quickly. If somebody's confused how to ask a question, there's an arrow in the top left, which is like a red arrow that you can press to open the menu where you can ask a question. I had a question about, are there any other parameters in the FAIMS device that can be used that would be interesting and can be changed besides the compensation voltage?
Yeah, so for actually what we have been testing, and especially for the phosphoproteomics, if we use different CV values, is actually to play around with the mass range, because for each CV there seems to be not only a selection on the charge state, but also on the different mass ranges that are where the peptides are gas phase fractionated. So one can actually optimize that such that you can optimize your windows for each of the CVs in a specific mass range that match the individual CVs.
So you can focus a bit more. Okay,
yeah.
Nice. Then I think we have some questions about just how to set up or how to select CV. So I know in our hands, the tipping point for using multiple CVs has been like around 1 hour. I'm not sure if that's also what you find in your experience with your setup, but in general,
that's also what we previously saw, at least. Yeah, but we actually quite intrigued now by this new data, especially from the 44-minute analysis, that it seems like that even 3 CVs seems to work better than just 2 CVs. But I guess, I mean, this is very fresh data, and we have to look a little bit more into it.
Right. Yeah. Okay. Yeah. So there was also a question about how to select a CV in case of longer gradients, and I guess it's still experimental, I suppose.
Yeah. So, exactly. So what we do is that we typically do like a CV sweep. So basically going from 30 up to 80 or 85 with 5 CV intervals, and then basically look at the overlap between the different CVs and choose those where you have the highest overlap or the lowest overlap, but the highest number of identified peptides.
Okay. Then we have another question. Does the detergent fractionation workflow you used work on tissues as well as cell lines?
We haven't tested it on tissues yet, but that is in the planning. But I cannot see why it shouldn't work. But I'll have to get back on that.
Okay. Also, what is the cleaning cycle for FAIMS in your experience?
Yeah, so I think we typically clean it if we run 24/7. Then we typically clean it once a week, and it's a very fast procedure. So it's it doesn't require more than half an hour or something like that from you take it off, clean it, and put it back on again.
Okay. Thank you for your presentation, and I think that's just about the time we had. So everything's on time, so we can now switch to the next presenter. Okay. Next up we have Florian Meier, who is now an independent research group leader at the Jena University in Germany. And Florian is also affiliated with Matthias Mann Group at the Max Planck Institute, and where he developed several new methods for data acquisition methods for quantitative mass spectrometry during his postdoc and PhD. And he was actually going to present 2 of his methods, namely BoxCar and DIA-PASEF, at today's presentation. At Biognosys, we had a long-standing collaboration with Florian on several topics, ranging from targeted proteomics using Max1 Live to DIA. I think if I were to mine my email inbox, probably Florian would be right there on the top of on the list of the most emails exchanged with external collaborators that I have had to do personally. So, and it's been a great pleasure, of course. I'm very excited to hear about his latest results with both of these methods and also with Spectronaut 14. And I'd like to hand over the mic to him.
Thanks, Tejas, for the very nice introduction. It's really been a great pleasure to work with you for the last years, and it's very nice have this opportunity now to talk about some of these projects. There's going to be a tour through several of these projects, but the overarching theme of the talk is going to be how can we handle ions efficiently as we shuffle them into our mass specs. So I see it a little bit as, you know, standing there in the rain and trying to capture as many droplets as possible with a little bucket. And actually these days with the brighter ion sources, it's actually not light rain, but it's really heavy rain. But still our bucket did not really increase. So we're still having the same bucket size to sample this rain. And this pretty much is the situation in Orbitrap MS1 analysis, where our bucket is the C-trap, and we have all the ions raining in from the ion source. And then what happens there in this situation is that we have this limited capacity in our bucket or in our C-trap, and we have to close the door after about 1 millisecond in general, traditionally in full scans in complex samples. So for instance, a HeLa digest or plasma digest, you really rapidly reach a charge capacity of the C-trap. And in our experience, that's about after 1 millisecond. And that's in stark contrast to the scan time that you need to resolve all of these ions by mass. So you would usually run at 60,000 resolution or even 120,000 resolution, which means that we would need a scan time of at least 128 milliseconds. So if you then do the math, you realize that we're actually using just 1% of the ion beam for our mass analysis. So to tackle this problem, we came up with the idea of the BoxCar acquisition method, where instead of letting all the ions enter the C-trap at once, we would use the mass selecting quadrupole to kind of chop the entire mass range into smaller windows. And this then gives us the chance to let ions in for longer times if they are low abundant and cut the ion injection times for very abundant signals. So in this picture here, you can imagine the blue ions to be very abundant. So you would inject them for just a very short time. Whereas the red ions, they are from an MRZ region that is apparently empty. So we can inject this region for a long time and accumulate this signal, for instance, for 10 or even 20 milliseconds as compared to the 1 millisecond before. So this gives us then what I usually call a fair ion count in the C-trap and gives us a better chance to see low abundant ions. Originally, we developed this method together with data-dependent acquisition, but when talking to the Biognosys team, we quickly realized that this could actually also be a very nice feature to data-independent acquisition. So we've been working on this for a while, and the main reasoning behind this was that our observation was that in normal DIA runs, we often have the situation that we see a very nice fragment ion signal. So we can be sure that the peptide of interest is there because we see its fragment ions. However, when we look at the MS1 scans at the very same retention time, we could actually not find any precursor signal that would fit to these fragment ions. So that's kind of a weird situation where you know that something is there, but you don't see it. And if we now look at the very same feature with BoxCar DIA, we actually see the precursor signal. So in this case, the precursor is depicted here in this with the yellow, red, and green traces for the different isotopic peaks. So the first advantage of doing this is that you, first of all, have your MS1 signal, so you can see your precursor ion, but it also helps you to define the boundaries of the peaks more accurately. So we've seen positive effects on the quantitative accuracy as well. And I think especially with the direct DIA methods coming up now more and more, I think this is a very, very interesting path to go for the future. So this is the way we implemented this. So we would still acquire a general or standard MS1 scan, but then after this, we would have these BoxCar scans, which are here depicted by these little boxes. And then after BoxCar scans, we would start filling the DIA windows, so the DIA segments, and cycle through all the windows that we have, and then start again from the full scan. So the big advantage of this workflow or this acquisition method is that you really boost your dynamic range on the MS1 level. However, as you can also see within this little figure, you add 2 extra scans to your acquisition cycle. And we found that especially in short gradients, this can actually be a hurdle, because you have to compensate now basically the dynamic range on your MS1 level with these extra scans. You have to compensate these with the number of DIA windows that you have, because in the end, you still want to have a good quantification, so your cycle time overall is limited to maybe 2 or 3 seconds. So there's just a limited number of scans that you can acquire in this time. So to overcome this problem, we endeavored in a new technology developed by Thermo Fisher Scientific, and it is super resolution Orbitrap mass spectrometry with a technology termed FAISDM, which stands for phase constrained deconvolution signal processing method. And there is a poster on this if you're interested in the details. What we did, the take home message is that this technology allows us now to essentially arrive at a similar mass resolution in about half the time. And of course, this is very interesting for short gradients, where we still want to have the high specificity of many DIA windows, but at the same time have the high quantitative accuracy of short cycle times. So this is just showing a few very first comparisons where we compared the standard signal processing, the EFT algorithm, to the FAISDM algorithm. And you can see that the benefit here for short gradients, I think this was 21 minutes EvoSep gradients, if I remember correctly. So with this in our hands, we then thought, okay, maybe we are now ready to tackle the problem that we are actually interested in for a long time, and which is really a long-standing topic in Tejas' lab, and that is tackling the high complexity and high dynamic range in plasma proteome. So I guess you're well aware of that plasma is extremely challenging for any mass spectrometer because of the very high dynamic range that we have. So we have these super abundant proteins like albumin that's basically everywhere, but at the same time we have all of these low abundant proteins, and we think that especially in these low abundant proteins there's a lot of potential to find new biomarkers because they're so notoriously hard to study. They have actually not been studied in great detail. So we think that this is actually a very interesting area to look for biomarkers, and we've been pushing the technology to go that far. And I'm really excited about a project that is spearheaded by Lily New from the Matthias's group in Copenhagen. And she really put all of these technological things that we developed over the last years together in one workflow and applied it to a biomarker study or discovery study in liver disease. And she will explain this in much more detail and much better than I could do in an oral session where you see the link down here. But I think it really shows that how far the technology is these days, and we're really in a position now where we can start applying our technology to find new biomarkers. So this was great. However, when I come back to this picture of the rain, there's also another problem. So it's not just that our bucket is rather small compared to all the ions coming in, but it's also that we have only one of these buckets. I think in the ideal mass spectrometry, you might have multiple buckets, and especially in DIA, you have the situation that you're only looking at a certain MORZ window at a time. So if we stay in this picture here, it would be that if you're running around with your bucket from one spot to another one, but you can only be at one spot at a time. And that's a bit frustrating because while you're at this one spot, you're basically ignoring all the other ions that are still coming down. And to tackle this challenge, we switched to another mass spectrometry platform, which is the Bruker QTOF platform, which has trapped ion mobility spectrometry in the front part. And together with Bruker, we developed this parallel accumulation serial fragmentation or PASEF method. And the idea behind this is that we make use of the trapped ion mobility spectrometry in the front part of the mass spectrometer to separate all the ions as they come into the mass spectrometer in time. So it's a bit like a stop and go system where we would trap the ions for a little while and then elute them one by one according to the ion mobility. And in the TIMS system, it's done in a way that the slow or low ion mobility ions with large collisional cross sections. So in this figure here, those would be the red ions. They elute first, and then the more mobile ions with smaller collisional cross sections elute later. So in this picture here, the blue ions would be the smallest ones with the highest ion mobility. And then the key of the PASEF method is that we make use of this temporal separation and synchronize the separation of the precursors in the TIMS with the precursor selection in the quadrupole. So in other words, as the red ions elute, we would switch the quadrupole to the MOVZ of these red ions. However, as soon as this mobility peak is over and the next batch of, or in the next ion mobility peak elutes, in this case the yellow ions, we would also switch the MOVZ selection window of the quadrupole. And what we achieve here, or the result of this, is that we are acquiring multiple ASMS spectra in the same time as we would have previously spent on acquiring just a single ASMS scan. And importantly, we do this without losing any sensitivity because the entire signal is compressed into a narrow ion mobility peak. And we're really excited about this technology, and you can hear more about this also at this ASMS. One talk that I would like to highlight is the talk from Andreas Brunner, where he's going to talk about how we can leverage this technology now to reach very high levels of sensitivity down to the level of single cells. So I can only recommend watching this talk. And this is kind of a live picture in the middle of a HeLa digest run, and you can see how this new dimension looks like. So on the x-axis, you have the MOVZ dimension that we are used to. Like the Z dimension would be in the back of the screen now. That would be the retention time. And then now what's new is this ion mobility dimension. And you can already see a few features here. So one of them is that the singly charged ion population, that is the one on top, is very well separated from the multiply charged species, which is the bigger cloud in the lower part of that panel. And the second feature that's kind of striking is that we see a clear correlation of ion mobility and mass. And for a long time, this was thought to be actually a downside of ion mobility, because it means that the separation technology is not ideally orthogonal. However, when we were looking for this for long enough, we were wondering, couldn't we use this also in our advantage? Having in mind that with the TIMS analyzer, we would scan this y-axis here basically in time. So you could imagine the mass spectrometer is seeing this heat map here slowly building up, going from the top to the bottom. And at any time or any single mass spectrum only sees one row of this two-dimensional heat map here. And we thought this should be especially helpful for data-independent acquisition, because it exactly overcomes this problem that I explained before, that when we are isolating a certain MOVZ window, we are not looking at all the other MOVZ windows. So the idea was here that combining the PASEF idea with data-independent acquisition would allow us to first look at the red ions here, but then as soon as the yellow ions elute, we could basically slide down with our quadrupole isolation window and still capture all of these ions. So we would no longer have to choose which ion we are looking at. We could simply look at all the ions. And to turn this idea or vision also into reality, we first teamed up with Hannes Rust from University of Toronto, Ben Collins, who's now at Queen's University Belfast, and Rudy Ebersold at ETH Zurich. And I'm really excited to share that we also published this now on bioRxiv. By now there's already version 2 of this preprint, and you can also have a look at the data. That's all going to be in there. So one big advantage of this ion mobility combined with data independent acquisition is that we can use now the ion mobility information to basically filter out peaks that we know are not related to our precursors of interest. It's really just using the ion mobility as a filter, and you can appreciate here on the right hand side the effect in signal to noise that you get from rather than extracting the full eye mobility window, just extracting a small portion of the eye mobility window, exactly where you expected this precursor. However, I think really the key difference and what makes this DIA-PASEF method so interesting is really the efficiency with which you can analyze the ions. So to bring this point home, I just want to go back to a very simple example. In this case, just a single peptide from a BSA digest eluting from a chromatographic column. So you could imagine as the peptide molecules come from the column, they would elute with a peak shape, something like this. And then when we look at the fragment ions that we would sample from this very peptide in data-dependent acquisition, we would most likely sample it somewhere at the beginning of the peak and actually turn just a very small portion of these ions into fragment ions. Then, similar to what Jesper said before, with this normal DIA method, this conversion is already much higher. So we're generating a much higher ion current on the MS2 level. However, with the normal DIA method, you cyclic through the window, meaning that you can only watch one window at a time. So your ion current somehow looks like this. But now having this stop and go function of the trapped ion mobility spectrometry in front, you can really tune your mass spectrometer to sample all the ions coming from the column. So you can reach up to 100% efficiency with our data independent acquisition mode here. And this works for simple digests, but also for more complex digests, as in this case in a HeLa digest, and it really gives us a handle to tune the sensitivity versus the selectivity. But importantly, no matter what we do, using this PASEF principle, we are always more efficient than with the standard DIA or even data-dependent acquisition method. So how does this look in reality? This is now a screenshot from the new Spectronaut version, which has really a nice support now for DIA-PASEF. And one window, one tab I like a lot is the visualization of the DIA-PASEF windows on top of such a two-dimensional heat map of the precursor signals. So this is very much the picture that we've looked at before, where we have these singly charged species at the top of the 2D map, so we can basically ignore them. And then what we are really interested in is this large population of the multiply charged precursors, which is where we think our peptides are. And these dashed and solid boxes, they visualize how we defined the window scheme. In this case, we used a total of 16 DIA-PASEF scans, and in each of these DIA-PASEF scans, we get 4 of these windows. This turned out to be very nice for a standard analysis, for instance, here of 200 nanogram HeLa digest with a 120-minute LCMS methods, where we achieved on average about 7,600 protein groups. And what was really stunning that even at this depth of the proteome, we achieve a very high data completeness above 98%. And it's also nice to see that we're not just deep, but we're also quantitatively accurate with a median CV below 10% and more than 6,000 protein groups quantified with a CV below 20%. And even more striking, it's not only working in our hands, but if you browse through the ASMS Reboot schedule and just type DIA-PASEF, you're going to find a lot of examples of applications already of DIA-PASEF. So be it on phosphoproteomics or especially tuned to short gradients. And I think I also have seen something about using direct DIA in the new Spectronaut version with DIA-PASEF. So I think this is really a very exciting development for the future. But I want to quickly go back in the last section of my talk to a more fundamental observation that we have had looking at of data. And if you analyze now your run in Spectronaut, there's this other nice tab that is showing the eye mobility calibration. So it's basically calibrating your empirical run to the library 1/K0 or the library eye mobility. And what was really interesting here is that when you look at this calibration curve, you realize that this is just a straight line. So it's, I don't know, if you looked at the same curve in retention time, you would see that this is hardly never a straight line, but it has some curves. But for the mobility, it really seems to be a straight line. And this is something we've also observed in a much larger data set. So in this example here, we acquired over 160 runs and compared the reproducibility of the retention time versus the reproducibility of the collisional cross-section values. And this slide is a bit busy, but what it actually shows is a matrix of pairwise comparisons between different runs. And then we were just calculating the Pearson correlation of either the retention time or the collisional cross-section in for the overlap of two runs. And as you can appreciate here from this comparison of retention time versus cross-section, it's with the retention time, you're much more likely to end up with batch effects. And in this visualization here, they turn out as patches of brighter color. And we could correlate these patches to changes of the chromatographic column, changes of the LC, or even from sample to sample. And we're really excited to see that we do not observe such effects for the collisional cross sections. And perhaps that's what you would have expected from a gas phase property, but it's really exciting to see that this also turns into reality. And what that means is that we can now start comparing collisional cross sections of peptides over long periods of time between instruments, between samples. And what we've seen in this large data set, looking at more than 300,000 unique peptides, is that we can still achieve coefficients of variation below 1%. I think this makes it a very, very interesting new dimension to proteomics, and we can learn a lot about these cross sections already from this data set. So, for instance, what has an influence on the collisional cross section? And you can see, even though it is highly correlated with the mass, there is subtle effects that would change the collisional cross section based on the linear sequence. So you could have the same mass, but a different collisional cross section depending on the physicochemical properties of the amino acids in your sequence, of the constitution of your amino acids, and also the position of specific amino acids. And this is really exciting. And of course, having such a large data set, having such a precise data set, we were also wondering if we could turn now linear peptide sequences into collisional cross-section. So we teamed up with the Helmholtz Center in Munich with a group of Fabian Theis and work led by Niklas Köhler to develop a deep neural net that would do exactly this job. So getting the peptide sequence in a charge state and predict the collisional cross-section. If you want to hear more about this, I have an oral presentation on this, and we also just published a bioRxiv preprint, I think 1 week ago, on this topic. What we learned from this endeavor is that, yes, we are able to predict CCS values just giving the linear sequence as an input. And currently our median accuracy is about 1.3%, and we achieve a very high Pearson correlation at above 0.99 across charge states and through the entire range of collisional cross sections. And I thought, because this is the Biognosys workshop, I was just curious whether we could actually use these predicted CCS values now also for our libraries in data independent acquisition. And so I really have to thank the developers of Spectronaut for making this new version so fast, because I basically started this analysis this morning, and I'm happy to already share the results. So they're preliminary, but I think they're quite exciting. And it's just based on a single raw file using the library that we also used before. And then if we use the experimental mobility values, we get around 87,000 precursors, which translates into 76,000 peptides, and from which we infer 7,500 protein groups. And then if we now replace the experimental values by the predicted values, I'm really starting to see that it almost has no effect or very marginal effect on the number of identified precursors. So it's really in a very similar range, and the number of protein groups identified with either the experimental or the predicted values is virtually the same. So with this, I would like to thank all the collaborators that are involved in all of these various projects, and my mentor, Matthias Mann, and the entire lab at the Max Planck Institute of Biochemistry, and also once more Biognosys for the great collaboration over the last years. Thank you.
Thanks, Florian, for the nice talk. Actually, I have a question regarding the deep learning collision cross-section part that you showed. So does this mean that you can use to predict animability annotation for libraries that you already had from other projects and use it to analyze your DIA data?
Yes, I think that's really one of the use cases that we now could basically take any library, maybe pan-human library that was acquired without an animability device, and we could now just in silico add this additional dimension to this.
Okay.
And I haven't tried it yet, but yeah, it would be very interesting to see how that works.
Okay. And what other kind of things you can imagine you could use this prediction for? I guess as a score potentially.
Yes, I think it's definitely an interesting thought to use this as a score.
Mm-hmm.
I'm curious to see how that works out. And I think the other theme that we've seen at ASMS last year, also this year, is just to have entirely in silico libraries, and together with the developments now predicting MS2 fragment ion intensities, predicting retention times. I think we now have a complete set where we could predict all the parameters that we would need for an in silico library, even for our mobility based work.
Still missing, but yes. Yeah. 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 I have a question here. Can BoxCar DIA method be used with FAIMS? It's kind of connect in the second talk.
Yes. Yeah. Excellent question. I actually have the very same question. I haven't had the time to test this yet and see how it performs, but in principle it should work. So the BoxCar DIA is implemented on the, I think, both the Explorers and the Eclipse platform. So the scan mode is there, and yeah, in principle one should just hook up the FAIMS device in front.
Okay. Then I have one more question. In terms of future perspective, where do you see the current limitations of DIA-PASEF, and where do you see the immediate improvements possible? If you have something to say about that.
Yeah, I think that the technology is still very, very new, and it feels like we're basically learning every day, and there's certainly a lot of untapped potential in this scan mode. There's basically an unlimited number of ways how you could define these boxes in this 2D space, which you use to select the precursors. So because now we basically place these boxes in not only in M over Z, but also in the ion mobility. And I think there's certainly different schemes that one could test, and maybe some of them provide better performance than what we have right now. Yeah, but it already works very well, I would say, for starting out methods. Okay, I think we are just over time, so we can wrap it up. Yeah, I would like to thank both Jesper and, of course, Florian. It's asking me for showing the screen again. Should I do that? Okay, good. Okay, for today's talks. It was very nice and really nice to see that Spectronaut 14 is already working quite well and in your hands. So yeah, very happy to see that. Then just to wrap up, just some information. We'll have another seminar next week on Tuesday with Birgit Schilling and Yansheng Liu. So that should be really interesting as well. So I hope
all of you join. And just general information, if you want to watch the recording of the Spectronaut 14 launch webinar that took place on Tuesday, or if you want to watch the recording of this webinar or the next one, you can do that at biognosys.com/ASMS 2020 reboots. Also, there's other information there. So yeah, with that, I would like to close today's webinar, and thanks everyone for attending, and thanks to our presenters. Bye. Bye.